Well, that's not helping well. That looks awful. Let me get the light on here. I'll move that closer too. That might help. This new Zoom doesn't seem to work very well. I'm really awful. Got the forest behind me, and that's the only part of the background I can see. And that's my green background too. Hmm. Oh well, I don't know what to do about that. So, can somebody please confirm you can hear me?
I can hear you.
Thank you. I've got about as bright in here as I can, and hmm, I don't know what to do about that. All right, let me share my screen. Let me state that I just finished grading the environmental part of the last practice quiz, and some of you need to work on showing me your environment. I've given you a warning if you're one of those persons. I've also complimented those who gave a very nice environmental scan, meaning they scanned the area around my computer. Why do I have that there? All right, before we begin, are there any questions? Come on. Not hearing anything, so let's begin. Today is July 22nd. Let me get my better glasses. I'm having a hard time seeing today. And we should be finishing chapter 3 and actually starting chapter 4. I don't know if we'll make that, but that's my goal. Do know that you have a quiz. It'll be your first real quiz tomorrow, this Wednesday. It'll cover chapter 1, chapter 10, the start of chapter 2, and then lab 0, 1, and lab 2. Any questions about any of that? Now, all quiz in this class, with the exception of the extra credit quiz and the plagiarism quiz, can only be taken one time. The quiz will be, the quiz questions will be pulled from the, what is that called? The objectives. But realize the objectives are general, so you have to learn a lot of material for each objective. Any questions about any of that? If not, let's begin with chapter 3. Is this where we had left off last time? Can somebody confirm that? Staining of microbial smears that we talked about?
Yes, that's what I have for the last one.
All right, thank you. That's what I remember too. Let me save that. So a dye consists of two parts. The chromophore will be in the colored portion, and a dye has a negative, a negative anion and a positive cation. And depending where the chromophore is, that will depend on whether the dye is a basic dye or an acidic dye. Because bacteria have a slightly negative charge, basic dyes are better for staining the cell. Crystal violet, methylene blue, malachite green, safranin are all basic dyes. They stain the cell. An acidic dye, on the other hand, stains the background around the cell. It does not tend to stain the microbe, or it stains them very lightly. Eosin, acid fusion, nigrosin, and what is it? India ink are acidic dyes. When you stain the background instead of the cell, it's called the negative stain or negative staining. And that refers to the old Polaroids where they would print out your picture and then give you a negative, and the negative would be the opposite coloring of a black and white photograph. Why it was called the negative. Any questions about any of that? And the negative stain stains the background instead of the cell. Why it's called the negative stain. Sort of like the old Polaroid negatives. Here we're taking a look at both simple stains. Let me blow this up. A basic stain, staining primarily the cell, and then a negative stain, staining the background. Both of them allow you to see the cell very nicely because they increase the contrast between the cell and the background of the slide. Any question about any of that? So this would be a basic stain like safranin, and this would be an acidic stain. I don't know an acidic stain that's pinkish or purplish, but India ink would be something like this, but India ink is black. When we're talking about stains and making a bacterial smear, you should realize there are three types of stains: a simple stain, a differential stain, and a special stain. A special stain is just a stain to stain a specific part of the cell. It has special purposes, like you want to stain the flagella. You use the flagellar stain or flagella stain, and it's a special stain. It's just a type of stain. A differential stain usually uses two or more dyes. That is not required, but they usually do use two or more dyes. And they allow you to see differences between different types of cells. A simple stain only uses one dye, and it will stain all cells the same, meaning they'll all have the same color or stain the same. So a simple stain uses a single dye. Its primary purpose is simply to increase the contrast between the background of the slide, that's the glass, and the cell itself. Doesn't matter if the simple stain is acidic or basic, it will increase the contrast between the cell and the background of the slide. The stain is applied to a fixed bacterial smear for a certain length of time. It may be washed off, it usually is. It's then dried and usually heat fixed and then examined under the microscope. And we'll talk about the different steps of making a stain or preparing a bacterial smear and then making the stain in a little bit. But do note that when you make a bacterial smear, it is not part of the stain. It's not part of the method of making the stain on the slides. This bacterial smear is something you do before you do the staining. A simple stain may have a mordant. A mordant is something that intensifies the stain. In the Gram stain, which is not a simple stain, when you apply iodine, that is a mordant. The mordant is not a stain. It increases the affinity between the stain and the specimen. It may coat the specimen to enhance it, thereby making it easier to see by increasing it. Like in the flagella stain, we have a mordant, and the mordant puts down layer upon layer. Let me blow this up. Of the mordant on the flagella, so that then you can easily see the flagella in the stain. Any question about any of that? A differential stain reacts differently with different kinds of microbes. Therefore, you can distinguish between two different types of microbes. A differential stain usually uses two dyes, but this is not required. Like with the endospore stain, it may only use, which is a differential stain, it may only use one stain. We're not going to talk about that stain, but we will talk about the Gram stain, which uses two dyes. The most commonly used differential stains are the Gram stain, which is actually the most commonly used stain in microbiology, and then an acid-fast stain, allowing you to see acid-fast cells, like Mycobacterium. The differential stain known as the Gram stain, which is the exact stain, was developed by Hans Christian Gram. He was actually a doctor, Dr. Gram. And when you spell out the Gram stain, you should capitalize the G because it's named after someone. And we always capitalize the first initial of somebody's last name, as well as usually the first initial of the first name. So the Gram stain should be capitalized with a capital G. It was developed in 1884. You don't need to know that. By Hans Christian Gram. He developed it to differentiate between the bacterial cells causing the infection and then the patient's own cells. So he did develop as a differential stain, but microbiologists soon began using his stain, and they noticed that it classified bacteria into two very large groups of cells, the gram-positive bacteria and the gram-negative bacteria. And I should mention that it also differentiates the archaebacteria into gram-positive archaebacteria and gram-negative archaebacteria as well. Any question about any of that? The gram-positive bacteria retain the purple color of the first dye, which is crystal violet, and the gram-negative bacteria lose the purple color of the first dye and then gain the color of the second dye, which is safranin in the Gram stain. And these are gram-negative cells, those are gram-positive cells. Any questions? All right, let's go over the overview of the Gram staining procedure. First, you make a bacterial smear on a glass slide. The bacterial smear is not part of the stain, so it's not part of the Gram stain. Then you air dry that slide and then heat fix it. You need to air dry and heat fix it to attach these cells to the glass of the slide. Otherwise, the cells will wash off, in the staining procedure. You then begin the Gram stain. First, you flood the slide with crystal violet, staining it for 60 seconds. Dump that off, wash the crystal violet with cold water, dump the cold water off, then flood the slide with iodine for 60 seconds. Iodine is a mordant. I'll talk more about that in a little bit. You then dump the iodine off, wash it once again with cold water, and then flood with acetone ethanol. That's the decolorizing agent. You apply the decolorizing agent for only around 5 seconds. And I'll explain this later, but you actually observe your slide while you're adding the decolorizing agent. So you put it at an angle like this. Can you guys see me here? Let me stop the screen and then I can see what the heck's going on. Stop sharing, I mean. Oh yeah, you can see that very nicely, actually. So that's nice. Sometimes I put it in front of my face because that's the only place where it shows up. And for some reason, the background isn't showing up, except for right there around the head of my hair and a little bit around the eyes too. Anyways, oh, there's the background there. It's like the green background isn't allowing the background to show up. That's weird. Oh well. I'm not going to try and figure out what's going on with Zoom, but you apply the decolorizing agent, and when the decolorizing agent starts running down the slide, you'll see the crystal violet streaking down the slide. When the purple stops running, you stop applying the decolorizing agent, and that's usually you apply it for only 5 seconds. Come on, share. You then wash the slide immediately with cold water. Dump the water off and then flood with safranin. Safranin is the counter stain. You apply the safranin for 1 minute to 2 minutes. And then dump the safranin off, wash with cold water. And then you blot dry and examine the slide under the microscope. So that's the overview. You'll note that after, whoops, go backwards, look forward. The staining procedure begins in step 3. But in the staining procedure, after every step, you wash the slide with cold water. Whenever you're giving me the gram stain, you only need to state one time, either the beginning, the end, or maybe even the beginning, doesn't in the middle, doesn't matter, that after every step you wash with cold water. You do not need to state after every step when you're listing the steps that after every step that there is a wash with cold water, meaning you don't just state wash with cold water, wash with cold water, wash with cold water, wash with cold water. You don't need to state it 4 times. You just need to state it 1 time after every step you wash with cold water. Any question about it, the overview? All right, let's take a look at the gram staining procedure. A little more detail than just the overview, where we're actually looking at the color of the 2 cells, the gram-positive cells and the gram-negative cell. First, you stain with crystal violet. All of the cells, gram-positive and gram-negative, take up the crystal violet. So they will be blue-purple in color. You then apply the iodine. The iodine will intensify the color of the crystal violet. So you note that the cells will be a little bit darker. That's not shown here. They'll also be a little bit different in color. It'll be more of a purple color than a blue purple color. That's not shown here as well. You then apply the decolorizing step, and that will pull the crystal violet out of the gram-negative cells, but not out of the gram-positive cells, as long as the decolorizing step is applied correctly. So here, the gram-negative cells will retain the purple color of the crystal violet. The gram-negative cells will now be clear. So this is the differential step of the gram stain, showing you differences between the gram-negative cells and the gram-positive cells. We then apply safranin, and it will color the gram-negative cells a pinkish color. The gram-positive cells will retain the purple color. Any question about any of that? You do need to know the colors of the different cells at each step of the gram stain. So you do need to memorize this slide or just learn it. So in the gram stain and grater procedure, first you make the bacteria smear on a glass slide. You do need to air dry it and heat fix it to the slide. Otherwise, the cells will wash off simply from adding the stain and then washing with the cold water. Any question about that? So step 1 and 2, that's something done before you begin the gram stain. And that's called the bacterial smear. Any question about any of that? You then apply the gram stain by flooding the slide with crystal violet for 60 seconds. And if you don't remember, crystal violet is a gram positive, is a basic bacteria with a positive charge, so the dye will stick to the cells. After 60 seconds, you then dump the crystal violet off and then wash with cold water. Crystal violet will stick to the peptidoglycan in the bacterial cell wall. Crystal violet is considered the primary stain because it's the first stain we apply, and it will color all cells the same color. So that both the gram-positive cells will be a blue-purple color, and then the gram-negative cells will also be a blue-purple color. Shown only purple here. You then, after the water wash, you then dump the water off. You don't need to get every drop of water off. You just need to get the excess water off the slide. You then apply iodine. Iodine is a mordant. It is not a dye. At least here, it's not a dye. Iodine increases the affinity of the crystal violet for the specimen. Iodine, when you're staining a photosynthetic cell, iodine can be a stain because iodine will stain the areas of a plant cell that have starch in them. It will stain them. Is it a black or a brownish color? I used to know this. Now I've forgotten. It's been so long since I've used iodine on plant cells. But when we're putting iodine in a bacteria cell, it is only a mordant. It's not acting as a dye. It will complex with the crystal violet to make a larger molecule than the crystal violet or the iodine alone. That will increase the intensity of the stain as well as make a larger complex in the cell. You want the larger complex because it will wash out of a gram-positive cells less easily than crystal violet alone. After applying the mordant for 60 seconds, you then dump it off, and then wash the slide with cold water. You wash the slide gently, but you wash it with cold water to remove the excess iodine. The color of both the gram-positive cells and the gram-negative cells will be a purple color. It will be slightly darker than the crystal violet alone, and it'll be a little more purple color than a blue purple color. Any questions about that? All right, after dumping off the excess water, you then apply the acetone ethanol agent, which is the decolorizing agent. It will wash the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells, as long as you're doing the decolorizing step correctly. And that is you're applying the decolorizing agent for only about 5 seconds. The best way to do it is to apply the decolorizing agent, watch the purple run out of the cells, and once the purple stops running out of the cells, you stop applying the decolorizing agent. So you just watch the purple running down the slide. And when it stops, you stop applying the decolorizing agent. You then need to wash the decolorizing agent immediately out of the cells by washing immediately with cold water. This has to be done immediately because traces of the decolorizing agent will be on the slide even if you dump the decolorizing agent off. And actually I had you having it at an angle so it'll run off. Because trace elements of the decolorizing agent can continue to pull the crystal violet Graham's iodine complex out of the cells. And if left on the cells long enough, the decolorizing agent will begin to pull the crystal violet iodine complex out of the gram-positive cells. Any question about any of that? So the decolorizing agent has to be on the cells long enough to pull out the crystal violet iodine complex from the gram-negative cells, but not long enough so that it starts pulling out the crystal violet iodine complex from the gram-positive cells. And that means the decolorizing step is tricky. It takes a little bit of practice. All right, invention, you wash the decolorizing agent immediately out of the cells to remove the decolorizing agent from the cells, because the trace amounts of the decolorizing agent will continue working on the slide. If you apply the decolorizing agent correctly, it will wash out the crystal violet iodine complex from the gram-negative cell walls, but not out of the gram-positive cell walls. Sorry, the gram-positive cell walls. And that means the gram-positive cells will be a purple color, and the gram-negative cells will be colorless. And so the decolorizing step is known as the differential step of the Gram stain. You then immediately wash the decolorizing agent out of the slide with cold water, dump the worst of the water off, and then apply safranin to the slide. Safranin is a pink dye, and it will stain all of the bacteria. We call it the counter stain because it has a different color to the primary stain. The primary stain being crystal violet, which is a purple color. Safranin is a pinkish color. You apply the safranin for 1 to 2 minutes. So in my procedure, it's very easy to remember because you're just applying all the agents for 1 minute, except for the decolorizing agent, which is only applied for around 5 seconds. After applying for 1 to 2 minutes with safranin, you then dump the safranin off the slide and then wash it with cold water. I've already stated that the safranin will make the gram-positive cells purple in color, and then the gram-negative cells pink in color. You then blot the slide dry with bibulus paper. Bibulus paper is very thick paper. Kind of looks like thin cardboard, actually. And you just blot the slide. So let's pretend this is the slide. Let me stop my share so I can see what I'm doing here. And then let's pretend this is bibulus paper. You rip the bibulus paper out of the paper holder. And then usually I fold it. And then I put it around the slide. And then just blot it down on the slide. Oops, you're not really seeing that. So you just blot it down on the slide. You do not wipe the slide with the bibulus paper or wipe the slide, meaning move the slide on the bibulus paper, because that will remove the cells. But if you blot it dry, meaning just push the blotting paper or bibulus paper onto the slide, it will provide enough contact with the slide to pull off the water. and you'll pull off enough of the water that you could actually apply oil to the slide and then use the oil under the microscope. You don't have to apply oil. You can just use the slide directly under the microscope. And if you blot it, you'll remove enough of the oil and water, you'll remove enough of the water to be able to use the slide under the microscope. Any question about any of that? All right, the gram-positive cells are purple after the procedure. The gram-negative cells are pink after this procedure. Why? What is the basis for the differential staining? And another question I have for you is, right here, I stated safranin is a pink dye, and it stains all the bacteria. So why are the gram-positive cells staining purple? If we applied safranin, and the safranin is staining the cells. Anyone have a guess? I don't expect you to know, but I thought somebody may get a guess. I'll actually give you extra credit if you get it right. When you stain a cell, so let's say this is our cell here, with the safranin, and you look at it under the microscope, or with your eyes, you will only see the purple dye, the crystal violet iodine complex. It's because crystal violet iodine is a darker dye, and our human eyes can only see the crystal violet iodine complex, meaning they can only see the purple. But if we were to look at the cell under the electron microscope, we would note that this area of the cell has pinkish color from the safranin, and this area of the cell has purplish color from the crystal violet iodine complex. So the only reason why we're not seeing the pink is because purple is sort of a dominant color, and the human eye can only see the purple. The pink is actually hidden from the purple, at least to the human eye and to the light microscope. To see the pink, we have to look at the cell under the electron microscope, but then we will see pink color in the gram-positive cell that has been stained with the gram stain. You will also see purple in the cell because it's a gram-positive cell. Any questions about any of that? In the gram-negative cell, we'll see no purple in the electron microscope because the crystal violet iodine has been washed out of the cell, but it will stain pink because it's applied after the decolorizing step. So what's the basis of the differential staining? Why do the gram-positive cells retain the purple dye or the crystal violet iodine complex, and the gram-negative cells lose the crystal violet iodine complex and then stain pink from the safranin? The basis is due to the differences in the cell wall structure of a gram-positive and a gram-negative bacteria. The gram-positive bacteria has a very thick peptidoglycan layer. The gram-negative bacteria cells have a much thinner peptidoglycan layer. They do have peptidoglycan in their cell walls, but it is thinner. The gram-negative cells also have a lipopolysaccharide layer that we'll talk about in a little bit. So the gram-positive cells shown here, let me blow the slide up, have a very thick cell wall shown in purple, which stains with the crystal violet iodine complex, meaning retains it in the gram stain. And then when we look at the cell wall, we'll note that it has layer upon layer of peptidoglycan, which is shown here. We're only seeing 3 layers of peptidoglycan in the gram-positive cell wall. There are at least 30 layers of peptidoglycan, not 3. In the gram-negative cell wall, here we see our cells stained with the gram cells, and it does have a peptidoglycan layer shown in purple, which actually doesn't stain purple with the gram stain. And then we have a yellow layer underneath it, which is the cell membrane, which we see here. And then we have a yellow layer above that cell wall, and that is known as the outer membrane of the cell wall. So this lipid layer is a part of the gram-negative cell wall. It's called the outer membrane of the cell wall. And let me blow that up a little bit. You'll note that the outer membrane of the cell wall largely is made up of a molecule that we find in the cell membrane, and that is the phospholipid. In fact, this layer of the outer membrane, it does have some carbohydrates and some proteins in it, but it's mostly phospholipid, the same as the cell membrane. And that's that layer of the outer membrane. This layer of the outer membrane is a little different. It does have phospholipid in it. Phospholipid is actually the main molecule in the, I guess, outer layer of the outer membrane of the cell wall. But it does have another lipid, lipid A, this blue blob here. And lipid A has an O polysaccharide attached to it, making this molecule a lipopolysaccharide. And that's only found in this layer of the bilayer known as the outer membrane of the cell wall. Any questions about any of that? All right. Now this is showing you only one layer of peptidoglycan in the gram-negative cell wall. In reality, the layers are between 3 and 20 layers of peptidoglycan in the gram-negative cell. So it's more than just one layer of peptidoglycan, but it's less than the layers of the gram-positive cell wall, which, as I stated, is something between 30 and 100 or more layers of peptidoglycan. Any question about any of that? We'll talk about some of the other molecules in the gram-positive cell wall and in the gram-negative cell wall in a little bit. I've already talked about lipid A in the gram-negative cell wall. But here we're talking about the difference of staining, and that is the crystal violet iodine complex initially forms in all cells, the gram-positive and gram-negative. However, the crystal violet iodine complex, it's larger than the crystal violet or iodine alone, and it is retained by the gram-positive cells, but it is lost by the gram-negative cells in the destaining step of the gram stain. Decolorizing step, I guess we could call it. The acetone or acetone ethanol wash removes the crystal violet iodine complex from the thinner peptidoglycan layers. Any question about that? So the crystal violet iodine is washed out of the gram-negative cell simply because the peptidoglycan layer is thinner and cannot retain the crystal violet iodine complex. So the cells are colorless after the decolorizing step, the alcohol wash. The alcohol wash will also dissolve the lipopolysaccharide layer, meaning the outer membrane of the cell wall will dissolve in the alcohol wash. It's because it's a lipid, mainly a lipid, I should say. The gram-positive cells, on the other hand, crystal violet iodine cannot be washed out of the intact, thick peptidoglycan layer just because it's thicker, and the cells therefore retain the purple color of the crystal violet iodine complex. So here we're looking at some cells stained by the gram stain. There is a gram-positive rod, some gram-positive cocci, a gram-negative rod, another gram-negative rod, other gram-negative rod, a gram-negative vibrio, meaning a curved rod, and then a gram-negative spirochete, one there and one there. Any question about any of that? All right. The gram stain is not always clear cut. Sometimes some problems happen, and then what do besides pull out your hair? Like here's the gram stain, and you can note these cells look kind of pinkish, gram negative. On the other hand, these cells, and certainly those cells, look purple. So what do we do for reading this? Those look purple right there too. Excuse me, pinkish, purple pinkish, and these look purple purple, and those look purple pinkish. So how do we count this? Well, when you're doing the gram stain, you only do the gram stain in good regions of the slide, right here, and you skip reading the gram stain in the bad regions of the stain, here, and there. What's happening here is, and there, is that when we apply the crystal violet iodine, actually just the crystal violet, some of the crystal violet came out of solution in the bottle and precipitated, forming crystals. And then when you applied the crystal violet to the slide, some of those crystals came out, and that's a crystal of crystal violet. There's a smaller one, and there's another smaller one. And why these cells are darker right there and there compared to these is that the crystal violet from the crystal is going into solution and then coloring the nearby cells. So you avoid this region, a bad region, which has a crystal of crystal violet stuck on the slide, and then you read the regions of cells which are in a better area. Any question about any of that? There are some other rules with the gram stain. You get the most consistent results when the gram stain is performed on young growing bacteria. If the bacteria are older than 2 days old, you should not apply the gram stain because the gram stain may give you incorrect results because the cells are simply too old. But if the cells are 2 days or less, you then apply the gram stain. And if they're older, what you do is you just streak out the bacteria and get new cells that are less than 2 days old and then stain them with the gram stain. Some bacterial cells stain poorly, if at all. It's usually the Microbacterium. And the results of the gram stain are not as clear-cut as the theory suggests. It would become evident if you were doing the gram stain for your unknown project. Some of you would be pulling out your hair. Because like I said, the gram stain is a bit tricky. It does take practice. The main thing is you need to apply the decolorizing agent long enough to get the crystal violet iodine complex out of the gram-negative cells, but not long enough that the decolorizing agent starts pulling the crystal violet iodine out of the gram-positive cells. If you don't put the decolorizing agent on long enough, then some, or at least some, of the gram-negative cells will stain purple. They'll retain the crystal violet iodine complex. But then again, if you apply the decolorizing agent for too long, you will remove the crystal violet iodine complex from some, if not all, of the gram-positive cells. Any question about any of that? All right, the structure of the gram-positive and gram-negative cell walls not only affects the staining, it also is related to how well the cells respond to antibiotics. Like gram-positive bacteria tend to be easily killed by penicillin G, as well as cephalosporin and many detergents. And that's because penicillin G, as well as the detergents and whatever, get right into the cells and then damage the cells. Whereas the gram-negative cells are resistant to penicillin G, cephalosporin, and many detergents. The reason why the gram-negative bacteria are more resistant to at least these antimicrobial agents is the antimicrobial agents are blocked out of the outer membrane of the gram-negative cell. The outer membrane, found only in gram-negative cells, will keep penicillin G from getting into the bacteria cell and then damaging the cell. Any question about any of that? Now, that does not mean the gram-negative bacteria are harder to kill than gram-positive bacteria. You just need to use the correct antibiotics on gram-negative bacteria. Penicillin G is not a correct bacteria to kill gram-negative bacteria because it won't kill them because it cannot get through the outer membrane of the cell wall and then damage the cell. One particular antibiotic that can kill gram-negative cells is, oh geez, I just lost it now. I had it on the tip of my tongue. I can't remember the name of it. It'll come to me, but another one is ampicillin. About the third member of the penicillin family discovered can actually damage the gram-negative cell as well as the gram-positive cell. And that's because ampicillin can get through the outer membrane of the cell wall, whereas penicillin G cannot. And that's why ampicillin and penicillin G are different at killing the gram-negative cell. I just remembered the antibiotic that can kill a gram-negative cell, and it's streptomycin. Streptomycin is a fat-soluble antibiotic, and it goes right through the outer membrane of the cell wall of a bacteria, and then can get in the cell and damage the cell. Any question about any of that? All right, you're not going to be examined on special stains, especially the capsule stain, but you may need to use the capsule stain for your unknown project in the lab. So let me briefly talk about the capsule stain. It is a stain used for viewing the cell capsule. The capsule stain is a special stain because it allows us to view if the cell has a capsule or does not. Normal staining procedure will wash away the cell capsule. So if you were to look at these cells in the gram stain, you would not see any capsule because it would wash away. all you would see would be this cell on the background of the cell, which is a light purple color. And that's a very dark purple color. But if you use the capsule stain, you can see that the background of the cell is stained a light purple, and then the cell itself is stained a very dark purple, and then the cell capsule is not stained at all. So it appears clear. Here's the capsule stain using India ink. India ink is black in color, and India ink does stain the background of the cell, while the background looks black. It also stains very lightly the cell, the bacteria. You can note these cells look grayish in color, and that's because they are picking up enough of the India ink to take on a color, but they don't pick up much India ink, and so they're like grayish in color. And then the capsule doesn't pick up any of the India ink, and so it is clear in color. Any questions about the capsule stain? I was looking to see if I can find a cell without a capsule in this picture, and I'm not finding one. We do have a few, like right there, right there, right there. We do have a few capsules without cells in them. And what might be happening is the cell, we're looking at the cell at a higher plane, and the cell might be down at a lower plane. And then when we're looking at this, we are cutting through the cell capsule. So here we're seeing the capsule, here and here as well. But we're not seeing enough of the cell. We're only at the very top of the capsule. And so the rest of the cell is down here. And so here we're only getting the capsule. So that's all you can see. But if you were to delve deeper from this focus area, then we would see the cell, assuming that it's actually in this capsule at a lower level. All right, so India ink allows us to see the capsule or a special stain, and the special stain does not stain the cell capsule, but it does tend to stain the background of the cell, and it also tends to stain the cell itself, like India ink does stain the cell itself, but it may be a lighter intensity, which it is in India ink, compared to the background of the cell. All right, any questions about this lesson? If not, that's it for chapter 3. Let me begin chapter 4, unless there are questions.
I do have a question that's off topic real quick.
Uh-huh.
There were two things I had questions about. The first one was the voting on labs. I was just wondering when that was gonna be counted because we did that voting on week 1.
Yeah, you could do it up to week 2. I need to count it because week 2 ended last Sunday or Saturday, 11:59 p.m. No. Well, anyways, you can't vote anymore. But I will count the voting. I haven't done that. I haven't even looked at it.
Okay. And then I did have a question about uh the 1 second. Um oh that when we get something wrong on the labs and you give us feedback, you tell me what I do wrong, but not the correct answer. I'm assuming, is that because if we have a second try, you'll do it on the second
Yeah,
one?
I will give you the correct answer on the second try. as I did in the first week's lab. So lab 0, 0 and lab 01. I have graded lab 00 now for 2 tries, meaning 2 times. And on the first grading, I only told you that this question was wrong. I didn't give you the answer. On the second grading, if you got the question wrong, I will tell you the correct answer.
Okay, and then also if we maybe forgot or didn't get a chance to go back and do the lab for a second time, are you going to go back and say the correct answers for the first lab that we submitted?
Um, if you didn't make the correction, then I usually do not regrade those labs. But if you email me, I will get your lab 00 graded with the correct answers. Meaning
Okay.
you have to submit a second lab before I go back and grade that lab. So if you
Okay.
did not give do the lab, I will not go back and grade that lab. So if you want the correct answers, you'll have to email me.
Okay. And then just lastly, I did look at your syllabus over again, and are we the first class that is not allowed to do open book or open no notes during the court or during this course for quizzes?
No, why?
I was just wondering because in your notes, I know, or in the syllabus, it says that we are allowed open books, but I just was making sure.
Is that correct? I thought I
Yeah.
changed that out of the syllabus. Where is that in the syllabus?
Let's see. It says under um says clean your area of all external materials, books, papers, and other computers or devices. You are allowed to have and use your textbook and class notes, but realize that if you look more than 1 or 2 answers, you'll run out of time to finish the exam.
So that's on the syllabus, or is that in the exam itself?
Uh,
Where are you reading that?
Um, hold on, it might have been under, I screenshotted it, so I don't know. I'm not
All right,
currently in
well,
the,
email it to me because obviously that's something I need to correct in this class, except for the plagiarism quiz and the first practice quiz. Every quiz may only be taken 1 time, and you may not use notes or your book. Okay, everything is closed book. And
uh, I just have another quick question. I'll let you get back to teaching. I was just wondering since you have done a closed book, and you used to do an open book, what is the success rate for your students using that, uh, using no notes, if you don't mind me asking?
the success rate is about the same. You're talking about people passing the class.
Yeah.
However, the number of A's is a little less, maybe 10% less.
Okay. Okay, that was my question.
All right. I started using the lockdown monitor about a year ago to make the class more similar to the class um, that if you had taken the class in the real classroom, where when you take the exam, it is closed book. Now, I always had it earlier before I used Lockdown Monitor. I had it open book and open notes because I could not verify whether someone was cheating or not. But I made the time so that you only had time for answering or looking up about 2 to 3 questions. And I thought that was good enough. Okay.
Okay. You know Professor Roberto, or the one that you get notes from for your lectures sometimes?
Uh-huh.
Do you know that if he does open book or if he does the same?
In class, he has closed book exams. He has only in class exams. They're not online anymore. He did when COVID-19 was around, or first came out, but he doesn't teach online anymore. And I think this term he's not teaching at all. Probably next
No, he's not.
year he will not as well because he's the department chairman. If he is teaching, he's teaching one class at most.
Okay. Thank you.
All right. Like I said, if you show me where you read that from, I will change that because I
It's
thought I had changed it everywhere in the syllabus as well as in the quizzes themselves. And it's possible I've got a mistake, in which case I'll correct it if you can show me where that is.
it's under lockdown browser requirement document.
Oh, well, that's the lockdown browser document. That one I haven't corrected. Okay. All right. I didn't even know I was talking about the quizzes in the lockdown browser. I'll make a note of that and get that corrected, but that's not talking about how to grade the quizzes. So
And there's no chance of you going back to open book
no
for your
chance.
students.
The main reason is because the special needs students, some of them get 2 times the time. And when I use Respondus Monitor, I noticed some of the students who had 2 times the time were actually able to look up all of the questions in the quizzes. And I'm not going to ever go back to that because they just have too much time to be open book, open notes.
What do you mean by special needs? Like someone that's actually special needs?
Someone who's special needs students, sometimes they're given 2 times the time.
That's a little distasteful, but okay,
Well,
thank
that's
you.
why I made it more fair, and that is there is no now open book, open note. And even if you're given 2 times the time, you cannot look up the answer in the notes or the book.
Okay, thank you.
All right. Um, let's see, I've got the share on here. So let's go to our next chapter, which is, I believe, chapter 4. All right, this slide is showing you guys. Can you guys see this? Yes, you can. Uh, the the overview and the main points of chapter 4, the functional anatomy of prokaryote and eukaryotic cells. I will talk about the functional anatomy of prokaryotic cells. The anatomy of eukaryotic cells you should have had in your preliminary biology course. So I'm actually not going to cover that topic. It is in the notes and it's in the reading, but I'm not going to cover it because you've already had it. I will talk about one part of eukaryotic cells, and that is the endosymbiotic theory. But the remainder of the anatomy of eukaryotic cells, I expect you to already know. Any question about any of that? All right, so the major goals and the rough outline is know the terms of this chapter. What's a coccus, a bacillus, a spirilla? What's a strepto, like a streptococcus? Staphylo, like staphylococcus, diplococcus? What's a tetrad? What's a sarxinia? What's a vibrio? What's a spirochete? What is the glycocalyx? What is a flagella? What is an axial filament? What is a fimbriae? What is a pili? What is peptidoglycan? What is endotoxin? What is selective permeability? What is diffusion? What is facilitated diffusion? What is osmosis? What is active transport? What is tonicity? What is a plasmid? What are inclusions? What are endospores? And what is the endosymbiotic theory? Now you'll note we have briefly talked about an endospore and an endotoxin and a flagella before. I think it was chapter 1 or chapter 10. So this will be the second time we're talking about some of these topics. And we'll go into in a little more detail. And then tell me how gram-positive and gram-negative cell walls differ. We already covered that in the last lesson, but we'll go into it in a little more detail in this lesson. And then describe the importance of taxis in bacteria. Any questions about this overview and the major goals? Now remember, if you want to know all of the goals for this chapter, you need to take a look at the chapter objectives, which will be the first part of the study guide for chapter 4. So here we're looking at a slide looking at the difference between prokaryotes and eukaryotes. Let me blow this slide up. Prokaryotes, those are Greek word for pre-nucleus, pro, before, pre. Karyote, used as nucleus, but in rheology, karyotes means kernel, and when we say pre-kernel, we mean pre-nucleus. Any question about that? Eukaryote, Greek word meaning true nucleus. Literally, it translates to true kernel. But like I said, kernel we're using for nucleus. Prokaryotes, at least the bacteria, generally have one circular chromosome. It's not in a nucleus. For eukaryotes, we usually have multiple linear chromosomes, meaning they are not in a circle. Like with the X chromosome shown here, the X begins here and then ends here. And the chromosomes are in the nucleus of the cell. For bacteria, does anyone know, is the DNA associated with histone proteins? Anyone have a guess? It's either yes or no. So
No.
give extra credit on that one. Yeah, that's correct. The DNA of bacteria is not associated with histone proteins. Histone proteins are not found in bacteria. The DNA of eukaryotes is associated with histone proteins. In fact, let me give this as an example. I don't have a piece of string here, but the DNA actually wraps around the histone protein in the DNA in a chromosome. So chromosomes have both proteins and DNA, both in bacteria as well as in eukaryotes. But in eukaryotes, the main protein is a histone protein, and the DNA actually wraps around 2 times the histone protein. In prokaryotes, the cells do not have organelles. We're talking about membrane-bound organelles. In eukaryotes, the cells do have organelles, and by organelle, I mean the term membrane-bound organelle. A membrane-bound organelle would be something like the mitochondria, the Golgi complex, the nucleus, a transport vesicle, and that's a membrane-bound organelle. When I use the term organelle, I do not mean a non-membrane-bound organelle. A non-membrane-bound organelle could be something like a cilia or what the heck is that called? Flagella, there we go. Or I'm losing the name of that. The mitotic spindle would be a non-membrane bound organelle. And we won't talk much about the non-membrane bound organelles. I called non-membrane bound organelles cell structures, and that way I don't even use the term non-membrane bound organelle. And then I usually call a membrane-bound organelle just an organelle. But it is a membrane-bound organelle, meaning the organelle is surrounded by a lipid membrane. Peptidoglycan is found in the bacteria cell wall. If the bacteria has a cell wall, peptidoglycan is the main molecule in that cell wall. There is no peptidoglycan in the cell walls of eukaryotes. And remember, animal cells don't even have a cell wall. So eukaryotes do not have any peptidoglycan. Bacteria primarily divide by binary fission, where the cell simply splits down the middle, and then half of the cell becomes a new daughter cell, the other half of the cell becomes the other daughter cell. Eukaryote cells divide by mitosis, where the cell undergoes mitosis to divide into two. Any questions about any of that? All right, we're going to talk about prokaryotes, the cell shape and the cell growth arrangement. We'll talk about cell structures in prokaryote cells. We'll also talk about the movement of materials across the membrane, such as the cell membrane, which is really the only membrane in a gram positive cell. And gram negative only have the cell membrane and the outer membrane of the cell wall. Eukaryotes, we're not really going to talk about them. We'll briefly talk about a few cell structures in eukaryotes, and then we'll talk about the endosymbiotic theory of eukaryotes. And that's the overview of this chapter. All right, when we talk about prokaryote cells, they have a size and a shape. The average size of a bacteria is between 1 and 2 or 3 micrometers. A very large prokaryote cell. Let me think, I used to know that. It can actually get up to, oh no, that's a virus. I'm not sure what the largest prokaryote cell is, but it's probably around 10 micrometers. They did discover a prokaryote cell recently that was very large, probably even larger than 10 micrometers. And initially they thought it was a eukaryote just because it was so large. But it's actually a prokaryote, and we won't talk about that. Very unusual. I think it's a bacteria. There are a basic shape to bacteria. In fact, all prokaryotes have a basic shape. There are round cells that we call coccus. There are rods that we call bacillus, and you may use the term rod interchangeably for bacillus. And then there are spirals that we call spirilla. A spiral is something like an S-like shape. And these are the three basic shapes of bacteria. Most bacteria, if they're not a coccus, a bacillus, or a spiral, they will be some combination of one or multiple of these three shapes. Like the spirochete is a spiral that has multiple spirals. And then the coccobacillus is a shape between a coccus and a rod. So this one here, it has a rounded edge. If this had a rounded edge and you make that rod shorter and shorter, eventually you would get a coccobacillus because you couldn't tell for fact whether it was a coccus or a bacillus. It's sort of in between, a coccobacillus. And then if you were to make it even shorter, it would be a coccus. Any question about any of that? And let me end the show here and jump ahead, stating that the author has this shape in the wrong place here. Not there, the next slide. Here we go. He's talking about the prokaryote cell arrangements. This is the cell growth arrangement. He's got the Coccobacillus in the cell arrangement. It shouldn't be here. Coccobacillus is not a cellular growth arrangement. It is a cell shape. And the Coccobacillus is like that. Let me show you this. There's a Coccobacillus. There's a Coccobacillus. There's a Coccobacillus. Two Coccobacillus. That one's about to divide or has divided. That one looks like it's so short. That actually looks like a coccus. But these are Coccobacillus. Any question about Coccobacillus? It is a shape between a coccus and a bacillus. A Coccobacillus is not a cell growth arrangement. So here it shouldn't be there. It should be in another slide. Such as here. Any questions? All right. There are different types of spirals. There are 3 types according to this slide. There's a vibrio, which is a half a spiral. You could also call it a bent rod. So a vibrio, you could either call it a spiral or a bent rod, either one. Any question about a vibrio? And there I got a curved rod there. There's a spiral, such as a spirillum, S-like shape here. I guess these are actual electron microscopic images, yeah. And then there's a spirochete. All it is a spiral with many spirals in it. The two are a little different in that this cell is more rigid, this cell is more flexible, and this cell can actually bend. We'll see a video where that cell, like this, can actually bend like that. I don't know if you can see that, but can actually bend like that. So this cell is more flexible. There are some unusual shapes of some prokaryotes, like the Stella bacteria, actually have a star shape. Shown here, this is the artist's rendition. This is an actual transmission electron microscopic image of 2 bacteria that have a star shape. And then there's the Haloarcula, which some people call square archaea. And if you look at that, that's not really a square, that's a rectangle. But it's unusual because, well, it's a rectangle. And a rectangle is not our 3 basic shapes of prokaryote cells. And then there are some bacteria which are pleomorphic, having almost any shape there. That sort of looks like a rounded rod. That looks like a dumbbell. I don't know, that's a Coccobacillus, I guess. No idea what shape that is. Or that one. That one looks like, I don't know, 2 eggs connected by something. Most bacterial species are monomorphic, where the cells have a single shape. Like all of these are square. Let's go back. All of these are vibrios. That one's almost a spiral, but not quite. All of these are coccus, all of these are rods, meaning they're monomorphic. But some species are pleomorphic, where the cells take on many different shapes, and I guess different sizes as well. Usually the cells which are pleomorphic are at least the bacteria, are the bacteria that lack a cell wall. There is one family of bacteria that lack a cell wall. The Mycoplasma is a family of bacteria that lack a cell wall. And the Mycoplasma tend to be pleomorphic in their shapes of the cells. So the cell wall tends to give the bacteria its shape. Any questions about any of that? All right, let's talk about the cellular growth arrangement. For the cocci, they have the most number of cellular growth arrangements. Shown here, and maybe shown right there. Let's blow this slide up. Shown here and there. These are singlets, a single bacteria. And it's a cocci, or a coccus, because we're only talking about one. And I guess I forgot to mention that a coccus means one. If you have more than one, it's plural, and cocci is the correct term. And actually, cocci can be pronounced 4 different ways. Let's see if I can remember all of them. There's cocci, there's cocci, cocci, and cocky. Those are the 4 correct ways to say cocci. So there's actually 4 ways you can say it. It does kind of depend on where you are in the world, where which pronunciation you use. Like they used to break it down in the United States. There's at least 3 regions of the United States. There's the western side, the eastern side, I think the southern side, and I don't know what the northern side uses, probably the western side. And I use the term cockeye, but you could call it cocky, as well as coxy or coxi. Yeah, there might even be a 5th way. I don't remember. Any pronunciation is correct. The point is, cocci is plural, coccus is singular, bacillus is singular, bacilli is plural. Any questions about that? With the bacilli, if you get that confused, just call them rod, a singular, and rods with an S, plural. Anyways, cocci have the most cellular growth arrangements. There is a singlet, a single coccus. There's diplococci, that's 2 cocci stuck together. There are streptococci, where there's multiple cocci stuck together in a string. There's a tetrad, where 4 cocci are stuck together. And you can see here there's a specific plane arrangement for how the cell divides to get the tetrad. And then there's a specific plane arrangement to get the cell to divide to give the shape of a Sarcenia. A Sarcenia is 8 cells stuck together. It's very hard to see the 8 here, especially when they're so crowded like that. But right there, you can sort of see 1, 2, 3, 4 cells of the tetrad stuck together. The 8, there's just too many of them, too many cells stuck together. And then you never see the one, like even here, you can't see the 8th cell because it's right there. And then cocci can also be a staphylococci, shown here. Staphylococci are the cocci in a cluster like a grape. Like if you put a stem there, this would look like a cluster of grapes. And that's actually what staphylococci means. It means cocci in a cluster like grapes. Any question about any of that? I guess we'll just go to the next. Bacilli have fewer cellular growth arrangements. There's a singlet, a single bacillus, shown here. There's diplobacilli, where they're connected together, 2 of them. There's streptobacilli, and this is where they're connected together on the short end, sort of like a sausage link. If you've ever seen sausage linked together this way, and that's called streptobacilli. And then there's the bacilli linked together on the long end, and that's called the palisade. Let me end the show here. Those of you who read Mark Twain will remember that Mark Twain was painting a palisade fence. The fence had a board of wood put together like this. And this is the ground down here. Anyone read Mark Twain? You remember Mark Twain was painting the palisade fence, and I believe he was trying to convince Huckleberry Finn that it was fun to paint it, and the reason was he wanted Huckleberry Finn to paint the fence for him. Anyone remember that? Nobody seems to. Erasing.
I never read it.
Okay. Anyways, the palisade fences aren't very common anymore, but they were very common in the past. And all they were with the piece of wood, a board of wood, where you stuck it this way, and then you made the fence like that. Sometimes you can find a palisade fence around a house like this, but so you stick it this way, meaning this is the soil, and there's the palisade fence. And those are the only cellular growth arrangement that bacilli have. There is a cell that has fewer cellular growth arrangements than a bacilli, and that is the spirals and the spirochetes. The spirochetes only have one cellular growth arrangement, and that is a singlet. All the spirochetes, whenever they divide, they do not stick together, and they only have single spirochetes. Any question about any of that? All right, there are two things you should note here. Streptococci means cocci stuck together in a string. Streptococcus with a capital X and then ending with coccus is a family name. So don't get them confused. Streptococcus is a family of bacteria, like Streptococcus aureus and Streptococcus epidermidis.
Is that one the strep throat?
Whoops, I did that wrong, didn't I? That's Staphylococcus and Staphylococcus epidermidis. Yeah, actually, Streptococcus is for strep throat. Streptococcus pyogenes is the species that causes bacteria. Sorry, I'm talking off the cuff of my head, and I got that wrong. So Streptococcus is the family, which includes Streptococcus pyogenes, which causes strep throat. Thank you for the correction, by the way. Oh, and then the other thing I need to talk about is the, where the heck is it? Oh, no, the bacillus is both the shape of a bacteria and the family name of a bacteria. Let's get a new one here. So if ever I say bacillus and you do not know, am I meaning the shape it should be lowercase b for the shape. it should also be regular type, meaning regular font. If on the other hand, I say Bacillus with a capital B, it is the family called Bacillus. And this should be either in italics. Let me blow this up so you can see it. Kind of see that it's Bacillus. Or you should underline it to indicate that it is a family name. Any questions about any of that? So it should be capitalized in either in italics or underlined if it's the family, Bacillus. And if it's the shape of the bacteria, it's lowercase b and regular font. Is that what you call that? Any questions about any of that? So don't get them confused. All right, let's talk about the structures of some typical prokaryote cells. We're taking a look here. All of the structures shown in red are found in all prokaryote cells. All prokaryote cells have a cytoplasm, they have ribosomes, they have a plasma membrane, also called the cell membrane, and they have a nucleoid region which contains the DNA. Note that the nucleoid region is not separated from the cytoplasm. The nucleoid region is wherever the DNA is floating in the cytoplasm. And there's no distinction between the cytoplasm and the nucleoid region, meaning there's no cell membrane or no membrane dividing the nucleoid region from the cytoplasm. In a eukaryote cell, we do have a membrane, the membrane of the nucleus, dividing the cytoplasm from the nucleus or the DNA. Any question about any of that? So for all cells, we have three components. You have the cell membrane or the plasma membrane. You have DNA, so that's found in all cells. And you have cytoplasm that's found in all cells. Ribosomes are found in all cells, but do realize that the ribosomes of eukaryotes are larger than the ribosomes of prokaryotes. A eukaryote has an 80S size of a ribosome, a little larger than the prokaryotes, which are 70S in size. And I think I talked about this before. The S just stands for the sedimentation coefficient, where the ribosome sediments when it is spun in an ultracentrifuge gradient. Other components of the prokaryote cell can include the capsule, that is a region of the cell secreted outside the cell, but it is secreted by the cell, but it is outside the cell. It's usually thicker than this, and we'll talk a little bit more about that, but sometimes it's kind of thin like this. And then there's the pilus, also called the sex pilus, or sex pili, or pili, if it's outside the cell. Pilus is singular. Pili is if there's more than one. There's also fimbriae, the hair is shown on the outside of the cell. There can be inclusions. That's something inside the cell that is accumulated to such levels that the accumulation causes the material to precipitate out of solution. And then there's this precipitate of the material in the cytoplasm, which you can see under the microscope, and we call it an inclusion. There's also the cell wall, It is external to the cell membrane, shown in yellow, also called the plasma membrane. Most bacteria do have a cell wall. If the bacteria has a capsule, it's outside the cell wall. There can be flagella. If the cell can move, it has at least one flagellum. That's singular. Flagella means that there's more than one. It's plural. As mentioned, the bacteria cell can have DNA. It usually does, otherwise it'd be a dead cell. I guess red blood cells don't have any DNA, but that's because they lost it. And when they're dividing, and then the cell matures, you lose the DNA. But bacteria cell can have the DNA in the chromosome that contains most of the DNA, most of the genes, but the DNA can also be in a plasmid. This is a small molecule of DNA in the cytoplasm. The main difference between this DNA and that DNA is this is small, only has a few genes. This is large DNA, has many genes. And I think that's everything. Any question about any of that? This is the artist's rendition, and that's an actual electron microscopic image. There we have a inclusion, maybe one there too. This region here looks like the nucleoid region. It stains a little different, but it has no boundary between the cytoplasm and the nucleoid region. It just stains different because that's where the DNA is. And you can't really see the cell membrane, the cell wall, and the capsule on this cell. They're all stuck together. All right, let's talk a little bit about the glycocalyx, also called glycocalyx. Is that what we called it before? No, we called it a capsule. There are 3 types of glycocalyxes. A glycocalyx is something secreted by many prokaryotes. There's even a glycocalyx on some eukaryote cells. It's something secreted by the cell. It is outside the cell wall, so it is outside the cell. The glycocalyx is usually sticky. It does have molecules in it, but the main molecule is water. There are 3 types of glycocalyxes, also called glycocalyxes. The first type is a capsule. The second type is a slime layer. The third type is an extracellular polysaccharide. The capsule is a neatly organized structure outside the cell wall. It does prevent phagocytosis of the bacteria by white blood cells. So the capsule can make the bacterium pathogenic, or at least more pathogenic, meaning more likely to cause disease. The capsule does have other molecules other than water, but once again, the main molecule is water in the capsule. The slime layer is unorganized and very loose. It is mostly water. So greater than 95% of the slime layer is water. The main difference between the capsule and the slime layer is the capsule can be viewed under the light microscope. The slime layer cannot be viewed under the light microscope because it is mostly water. To see the slime layer, you have to look under the electron microscope. Any question about any of that? The extracellular polysaccharide is found only in biofilms. And the cells, let me do a picture of that. You can have a cell right here, and another cell right here. You note I'm making different species here. Another cell, let's make it here. And they can secrete the EPS layer. And initially, this will be a capsule because you can view it. But as they continue to secrete the layer, it'll take up more space. Sorry, it's hard to erase this. I'm not doing a good job, but you're getting the picture. Let's do it there. As the capsule continues to grow, they then begin connecting together. And this is called the slime layer, which, not slime layer, sorry, the extracellular polysaccharide, which is in the biofilm. And this layer forms the biofilm that the cells are in. And this biofilm just continues to grow as the cells secrete more and more of it. I'm only going to do it on the top side. It's just too hard to erase all of it. Oh, wrong one. And it'll start incorporating more cells inside the biofilm. And that creates the biofilm. We're not going to talk much about biofilms in this class, but if you want to know, one biofilm that almost all of us have is the buildup of tartar on our teeth. Comes about from the bacteria creating a biofilm. And this biofilm grows and then hardens with time. So initially it's called plaque, and plaque is the biofilm. And then it grows very hard, and that we call the tartar. Any question about any of that? So the main glycocalyx, we'll talk about in this class, is the capsule. That is a glycocalyx you can see with the light microscope. It's neatly organized, has molecules in it, meaning besides water, and is something that prevents the cell from being phagocytized. The slime layer, on the other hand, is mostly water, cannot be seen by the light microscope. And the extracellular polysaccharide we'll almost never talk about in this class. Oops, I went backwards, I think. All right, the flagella is a structure found on the cell. It is mostly external to the cell, but there is a small portion of the flagella inside the cell wall. That's this region here. There's actually a very small portion of the flagella which is inside the cell membrane, the plasma membrane. So most of it is outside the cell, but a little bit of it is inside the cell wall and even inside the cell membrane. If the cell can move, it has at least one flagellum, either one flagellum or multiple flagella. Flagella are made of chains of one protein called flagellin. So the filament here is made of one protein called flagellin. Any questions about flagella? If there's no questions, I think we're about to end this class. Let me check on that. Sorry, it's the summer term. I don't remember. 6:50. Yeah, I think we'll stop early and I'll stop there. Any questions? If not, I'll see you at 7 o'clock for the lab. Yep, not there. All right, bye.